Hello everyone and welcome to our NGS-CN Summer School online collection on spatial transcriptomics. Before we jump to our next speaker, let's take a look once more who we are. So it's a next generation sequencing competence network that consists of 4 production sites as you see on the map. So in 2019, DFG has funded centralized sequencing applications. There were quite a lot of calls, and within each call there were a lot of projects. And from 2024, DFG has changed the strategy towards individual sequencing applications. That means that now there is no special deadline for a centralized call, and you can co-apply at the DFG together with the chosen genomic facility. If you wish, here is a QR code, contact us, and then we could take a look and maybe sequence together. If you want to know something more about our news and upcoming events, then you also can take a look at the QR code, scan it, and get into our mailing list. Let's grow community together. And with that to be said, let's welcome our next speaker.
So thank you for the introduction. Again, I'm Joana. I work with Philipp Rosenstiel in Kiel. So we are in a competence center up here in the north. I just also wanted to have a disclaimer today. So my title is Integrating Single Cell into a Spatial Context. So I do a lot of single cell, and only now with the tissues that we work on, we'll be able to start to acquire spatial transcriptomics. So it won't be a lot of beautiful images of intestinal epithelium, but it will be a lot on how we are using single cell omics and validating our findings into a spatial context. So in particular, in personalized medicine. And this is how we start as well. So I would like to first introduce to you the type of diseases that we studied. This is just, if you're squeamish, please don't look now because there will be images of human. And so we are normally studying chronic inflammatory diseases, in particular Crohn's disease and ulcerative colitis. We have this umbrella term of CID, so chronic inflammatory diseases. These are diseases that are characterized by this exacerbation of inflammatory response that cannot be stopped. They can occur everywhere in the human body, and they can also occur at different times, so lifetime points. So they are very heterogeneous, and that makes them really hard to study. So this definition of a complex disease, because not only they have genomic contribution factors, so things related to the genes and so forth, and they're heritable and pass through your family, but there are also a lot of environmental triggers for these types of diseases, like smoking, diet, etc. So it's very difficult to study what is a cause, but also how do we treat these types of diseases? That is mostly what we are interested on to study in our group. We do have a lot of omics layers that we normally use to study these types of diseases. Today I will focus more on the transcriptome, but we normally also have information about the metallome, about the microbiome, whether it's 16S and nowadays also metagenomics. And we also have metabolomics, etc. So a lot of different layers of information that aids us to study this type of diseases. But starting with the transcriptome, so on the right side is our all 4 techniques that we already have been successfully using to studying CID. So of course, many years we've been using bulk RNA-seq. So these are transcriptome data and transcription profiles coming from tissues. Of course, the greatest advance in the last 6-8 years has been the single-cell RNA-seq, which also we've been successful into using. We also have some projects with single nuclei where we do, like, for example, brain approaches and also study of adipose tissue. And now we're really starting with the spatial transcriptomics because our tissues are so difficult to manipulate. And what are the questions that we tend to ask? So for the treatment of the CIDs, one thing that we can ask is just very general, what is like the gene expression of a certain disease? So disease-specific gene expression changes. What is ulcerative colitis compared with healthy? What is SLE compared to healthy? Things like that. So very simple questions. Another question that we can ask from here is also to not only know the genes that are affected, but then trying to put them into context. So enrichment analysis and molecular pathways. But normally we are not so much involved in the diagnosis of the disease, but we are mostly involved in a type of prognosis and in particular treatment response. So in our group, we really deal with chronic inflammatory that has very high activity, and they are treated with biologic treatments. And we are really interested in studying treatment response. So once you've been diagnosed with a disease, how can we make sure that the treatment that you have is the correct one that will lead you to a disease control, so a decrease in the burden of your disease for as many years as possible. So just changing gears a bit here. So why do we do so much single-cell transcriptomics, and why is this presentation about single-cell transcriptomics? Of course, we, as many of us before, have done a lot of bulk RNA-seq, but because of the nature of the disease that we study, this immune part is so prevalent and sometimes can be masked in the tissue. So we are really interested in having the granularity that single-cell RNA-seq gives us. So to identify the individual immune cell types, so macrophages, B cells, megakaryocytes, etc. And what is in particular with the cell types? So what are the inflammatory pathways that are altered before and after treatment? So I normally like to use the analogy. I ask, why is the smoothie sour? So why do you have a disease? And if you do bulk, you cannot really know. But if you do single cell, I can tell you which type of fruit is actually causing the smoothie to be sour, which in this case is like, oh, is this particular phenotype of B cells that making this exacerbation inflammation, just as an example. And nowadays we can have another layer, which is, I call it the fruit pie. So we can not only tell you that the important point is the B cell, but because these diseases are so specific to the tissue that you find them, I can also tell you it's B cells because of this particular context. So this is what spatial is giving us right now, this layer of information. So we start slowly. We start with like the bulk RNA-seq analysis that we have been doing so far. So here we ask the question from a normal person suffering from ulcerative colitis, so this inflammation of the intestinal tract, are we able to predict if the patient is going to have a flare? And this flare means they're going to have an increase very rapidly in their disease burden. So can we actually predict this based on a tissue sample? And this is some of the model approaches that we use, of course, because we work with patients' data and we also work with patients' data longitudinally. We always have to have this information into our models. We also here, for example, have the information of sex and age group, so it's not confounded. So that's exactly the question that I asked. So I wanted to identify particular genes that are kind of related to flare. So on the right side, you can see a very big heat map with some genes. And what is one? So one is actually this particular case of patients that we are interested. Each of these lines is a sample. And we are interested in these patients that seem to have very normal state. So they are not undergoing any type of inflammation or beginning of inflammation. However, we know that soon future, and this is within a year, there is going to be a development of the disease, and these patients are going to have like systemic inflammation of their colon. So I'm trying to identify these particular genes, and then I identify what these particular genes do. So this is what I asked. So we could see that there was an upregulation of genes that are related to endothelium development, to leukocyte-mediated immunity, and also to B-cell immunity. And we do think this is really interesting because then we can take this information, we can relate back of what we know so far, and we can also try to validate in, for example, immunohistochemistry sites that we have available. So we validated in an external cohort that there is potentially this mesenchymal and this fibrosis that is already inherent in the tissue. And if you reach a peak on this fibrosis state, there is a chance that you're going to have dysregulated inflammation soon enough. So we are in the late stage of trying to validate this. We are not so now interested in this indication that there is also an effect of B-cell mediated immunity. This is a little bit more difficult to capture because we believe that this is connected to the lymphoid vessels that you find in the intestinal epithelium. And those are not always captured once you take a sample. So you really have to kind of take multiple samples to be able to see that there is this increase in the number of B cells. So we're trying to find new ways to try to kind of develop our insights from bulk RNA-seq into the spatial context. So I will present a bit more going forward. The second kind of approach that I want to talk to you about is using single cell. In this case, it's a little bit different approach. So here we are interested in really therapy response. So we have time points that we collected biopsies before a treatment is given, and then 2 weeks, 6 weeks, and 14 weeks. So the 14 weeks point is normally when we take another biopsy, so baseline in 14 weeks. And here we try to compare what is changing between these 2 points of before and after treatment. So of course, here you can do different types of approaches. You can do pairwise model comparison, which is compare before and after. Also very keen on doing co-expression analysis with single cells. So you can really see like groups of genes that have similar patterns throughout the treatment. And this is also what we did here. Today, I will only explain on the pairwise models. So this is a little bit on the cohort design. So we have both ulcerative colitis and Crohn's disease patients this time, and we have kind of two collection of samples. So per time point, we have before treatment, we have a collection of a sample of the inflamed and non-inflamed tissue, which is really cool. So we can kind of compare what it means to be inflamed to the non-inflamed within the patient. So it's like the perfect control. And then we have another sampling time, which is 14 weeks after the treatment was initiated. And again, if possible, we took also inflamed and non-inflamed samples. So this is a bit how it looks like. So of course, if the patient has non-remission, so if the patient is not feeling better, we only take a sample from the inflamed tissue because normally there's a spread, so you cannot take a sample again that is close by. So this is how our cohort looks like. This is just a subset of the cohort. So there's more patients that have been included now. I'm just showing you now a subset. So we have here 5 patients, 3 of them underwent remission. This means that even after 14 weeks, they start to respond to the treatment positively. So it means that they are feeling better, their inflammatory markers are down, and the patient themselves feel better than before. And then we have the non-remission patients, which is patients, they can still be responding to treatment, so they can still have a decrease in their disease activity, but they're not feeling like significantly better. So this is why they are called non-remission then. And as I said, we took intestinal epithelium. And if you ever work with intestinal epithelium biopsies, you might know that these are very difficult to deal with, but we now have a protocol, and we're also going to be publishing it soon. If anybody else is interested, I can share a preprint of protocols to actually process these types of tissues. So this is kind of like our first round of successful processing of biopsies. And now we have even better protocols, so you can actually acquire up to 12,000 cells per sample. So it's just a little detail here. So in this case, we were still not as good as I just mentioned. So we were able to identify 26,000, so almost 27,000 cells here. These are all done with 3' protocols from 10x. And there's also a bit of multiplex in this, but I will mention it another time. So you can here on the left side identify, I already did the cell type annotation, which is of course crucial. but you can already separate pretty well these immune clusters from the epithelium clusters, from the endothelium and lymphatic clusters. So I'm pretty happy with this cell annotation. Because of course, what you want is you want to have something that is also like reminiscent of what we know already from the sample that we take. So going forward, it starts to get a little bit heavy. We could see also that there might be some differences between CD and UC, especially once we talk about plasma cells, for example, and even enterocytes. So these are kind of the cell types that we are interested in. And because we have more Crohn's disease patients in this subset, that's also what I will talk more about. So first talking about the enterocytes, and this is the comparison at baseline only, comparing most inflamed biopsy with the least inflamed biopsy. We could see that there was this kind of down regulated genes in the most inflamed, which is kind of counterintuitive. We could see that these genes were related to copper and zinc stress response. And then we could already see, like based on previous literature, that is actually like positively related to the amount of to other inflammatory markers such as CRP and calprotectin. So there is this idea that there needs to be active inflammation that is related to the treatment response. So this is kind of what we are going for. Then we have the plasma cells. So these are plasma B cells, and we are more interested in comparing the baseline with the 14 weeks. Just a little like to tell you, there's a bunch of different treatments, and of course, these different treatments will affect the cell types differently. Now we have a bigger set, so we have more treatments here available, so we can try to disentangle treatment-specific responses. But for this moment, we don't have it here. I just want to talk a bit about the plasma cells. So there was at baseline, most of the genes downregulated, and then we have some genes upregulated. Talking about these genes, we could see that the genes that were downregulated and at the most inflamed were related to IgE-mediated immune responses, that's Fc gamma receptor. The upregulated genes were related to B cell receptor signaling. And of course, there's numerous literature on this. And we could also see that this was very much related to the non-remission. So this activation of Ig and Fc gamma responses are mostly related to the patient that have non-remission, which is kind of interesting because so basically in this case, the group of samples that changed the most was actually the non-remission patients. So they were not really able to control. The treatment affected the homeostasis of the tissue. However, it seems that the non-remission patients had like an exacerbated response, which made them not being able to, for the patient to feel better overall. So actually the signature is very much related to the non-remission. And what we are doing now, working together with colleagues from the Fraunhofer Institute in Hanover, we actually trying to validate some of these genes that we find, particularly in the plasma cells. So this is image of a multiplex immunohistochemistry cut of the intestinal epithelium, and they can do this for many more samples that we can process with single cell. So we are trying to be able to validate this. So this is more than going back to the spatial perspective with a few markers that you can stain in your multiplex immunohistochemistry, and also kind of, in a sense, going the other. So we did all the work in discovery, and then we find these markers and then give it back to the pathology department to see if with these markers they are actually able to say if the patient will undergo remission or not already at baseline, which is again what we really want to do, because we really want to know if the treatment that we are giving the patient will actually make him feel better and not worse, right? We don't want to give the patients this treatment if we know already that they don't work, that they do not work. So The last thing I want to talk about, and this is like my final project because this is also what I'm actively working on at the moment, is again to predict therapy response with a bigger cohort. So, and in this case, we also have single cell and spatial data. So this is a little bit of the experimental design. This is a project that is within the Immuniverse project of, so this is a EMI consortium from the European Union. And what we did is deep phenotyping of ulcerative colitis patients that are treated with either anti-integrin or a JAK inhibitor. And the approach is quite similar. So we have this lesional and non-lesional tissues at baseline, and then 14 weeks later, we also compare if there is, if we also take a sample of the biopsy, so a sample of the intestinal epithelium, we take a biopsy, and then we classify the patients if they are remission or non-remission. And this is not only done for ulcerative colitis, but in this case, we also have atopic dermatitis because in this chronic inflammatory disease, they also are given JAK inhibitors, which are the exact same medicaments that it is used for UC. So it's also quite interesting to compare what we see in the skin, which is, I would say, a easier compartment to access than intestinal epithelium, if we see the same patterns of cell type response to these biologic treatments. So again, this is what I'm really working at the moment. So the single cell object is now done, also with good cell annotation, good separation of stromal immune and epithelium cells. And what we are now doing is we are actually projecting these cell types at the spatial level. This is Visium HD samples. They are very similar. Of course, they were preserved differently, but they were taken at the same time for the patients that you just saw in the single cell. And this is what we are doing now. So we are actually able to put multiple biopsy cuts in one Visium HD because they are so small. We can do multiples. It's much better for us because we are not losing the whole information just because our biopsies are very small. So yeah, so now we can kind of project what we learn, and we could also already see that, for example, for the introcytes, there's already some differences that we have seen previously. So MUC1, which is a goblet cell marker, so these are the secretory cells of the intestinal epithelium, seems to be affected differently before and after treatment. And also DUOX2, which is related to glycolysis, seems to be affected. So right now, I'm trying to run the integration approach, so to join the single-cell RNA-seq and the spatial already into one type of file that we can project and ask questions and also do some type of neighborhood analysis. So this is what I'm actively working now. Very exciting times, and I'm very excited to be able to talk to you about this, to have this also the single cell and the spatial information, and can we actually in the end try to make things better for the patients that we have at our hospital. So this is kind of where I want to leave you. I also was digging through my presentations that I had before with the NGS Competence Network, and I saw that I also had this that was done a long time ago for the summer school, that I had this slide. So you see it ends at 2021. So this is from 2021. And you can see here how we were able to acquire more and more single cells. Has the methodology become like it's been evolving. And now we are reaching another layer, which is like not only acquiring all these single cells, but we are also now putting them into context. So I try to see like how, so my question is also to my boss and to you is like, where is the limit? Like how many cells, how many transcriptome profiles, whether they are single cell or spatial, do we need to acquire? Do we need to learn to be able to create real-time effect in precision medicine? So this is something that I will also would like to leave you with this question, because this is something that I asked myself many times. So how much more effort do we need to put into this thing to try to translate things right now into the hospital? Do we need to focus on a more homogeneous population base and only focus on Northern German population? Because of course I'm here working in Kiel. Do we need to also more and more acquire heterogeneous data sets, people with different ethnicities, people that are elderly and just got uh, uh, diagnosed with IBD, for example, because they were not diagnosed before? So the more heterogeneous data sets. And will the heterogeneous data sets also give us information that we can potentially use in the clinic if we really want to go back to this personalization of medicine at the clinical level? I mean, I think we all agree that we, as it is right now, and with the price tags that we have, we cannot really use spatial or single cell transcriptomics for patient prognosis. This is only, I don't think, it's not only because of the amount of money, but also because of the time that it takes to process samples is not really feasible to do it at the clinic. So also trying to find different ways that we can validate what we find that do not require this in-depth omics layer information. I think this is also very important. Yeah. So who will pay if we actually start using spatial transcriptomics in a routine clinical? That's also a very good question. So this is what I want to talk to you about. So thank you everyone here, and we see next week. If you have any questions, please let me know.